So now we're going to continue on with molecular biology of the gene. We have talked about replication of the DNA, and so now we're going to talk about gene expression and how that relates specifically to genes on DNA. Okay, so last week we talked about DNA replication, and the purpose of DNA replication was to allow for cells to divide, and that can either be through mitosis or through meiosis. And so we're taking DNA and we are putting it into the next generation of cells. So today we're going to start talking about gene expression. And so the purpose of gene expression is to make proteins. So we've talked about DNA is made up of genes, and so gene expression is taking DNA, making RNA from the DNA, and then the RNA being read to make a series of amino acids, which of course are the monomers of our proteins. Okay, so let's talk about some proteins you've probably heard of. So do any of these look familiar to you? We've talked about several of them. CDK and cyclin, remember, were proteins that regulated the cell cycle. Actin and tubulin are part of the cytoskeleton. ATP synthase, we know we used in cellular respiration to make ATP. Insulin helps to regulate your blood sugar levels. Hemoglobin is a protein in your red blood cells that allows it to carry oxygen to various parts of your body. So all of these are proteins, and as you know, they're super important for any kind of metabolism within your body. So where do these proteins come from? Well, they come from genes on your DNA. So the definition of a gene, it's a specific sequence within the DNA that codes for a specific protein. I don't know why I just said proton. So, for example, when we want to produce an insulin protein, there is a gene called the insulin gene, which is a specific sequence of DNA that codes for that particular protein. So we know that DNA is made up of monomers called nucleotides. And so when we align those nucleotides in specific orders, they are like an alphabet that reads for, this is the protein I want to produce. And so it's like we need a decoder. So the mRNA comes in, and it is sort of like a complementary sequencing event of the DNA genes. And that mRNA is then read by other RNAs that we'll talk about to make the proteins. And the way they do that is the sequence of nucleotides gets converted to a sequence of amino acids. So the mRNA reads the sequence of nucleotides in the DNA and then produces the sequence of amino acids that then eventually make up the protein. Okay, so let's take a closer look at RNA for a minute. So some of this is going to be a review for you, but I do want to make sure that we know how to distinguish it from DNA. So RNA is a ribonucleic acid. Remember, the RNA comes because it's a ribose sugar, and that is in comparison to DNA, which is a deoxyribose sugar. So this one has its oxygen. This is a ribose sugar. So within RNA, remember when we're looking at our bases that you do not have thymine in RNA. Instead, you have uracil. RNA is single-stranded as opposed to DNA that's double-stranded. And the nice thing is it can be temporarily paired up with DNA using that base pairing rules. And remember, those base pairs are connected by hydrogen bonds. So one thing, if you'll remember from way back when we were talking about our organic chemistry, is that those hydrogen bonds are strong enough to hold them together temporarily, but they're fairly weak bonds. It's not a covalent bond. So they can come together temporarily in a weak bond in order to move forward with our protein production. So here is our structure of our RNA. It's going to be similar to if we took the ladder apart on the DNA. So if you look along the sides of the ladder are still phosphodiester bonds, right? So those covalent bonds that link the sugars together through the phosphate group. And then off to the side, we're going to have all these bases. And that's what's going to pair up with the DNA. And just note again that we have guanine, uracil, adenine, and cytosine. So no thymine whatsoever. So remember each one of these sections right here. So we have our pentose sugar, our phosphate group, and our base pair. Our base, blah, 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 can't talk. Let me try that again. Phosphate group, ribose, and then our base. That is one nucleotide. So our structure of DNA is multiple nucleotides linked together through phosphodiester bonds. So here's where it gets interesting. So DNA, we pretty much have one type of DNA. But for RNA, we have 3 different types. And that really has to do with its function, not with its structure. So we have mRNA, which I've talked about a little bit. That's our messenger RNA. That's the middleman between DNA and the production of proteins. Then we have what's called tRNA, or transfer RNA, and it has what we call anticodons, which we'll talk about, that carries amino acids throughout the cell. And then we have our rRNA, ribosomal RNA, and that's found in the ribosomes and helps position the mRNA within the ribosome. If you'll remember, the ribosome is where proteins are made. So it helps position that mRNA within the ribosome so the protein can be made. And just the other thing to remember is that the ribosomal RNA is basically the universal RNA. Remember, every cell has ribosomes, and every cell has ribosomal RNA, regardless of what type of cell it is. So ribosomal RNA has often been used when you sequence it to see how closely related organisms are. So it seems like ribosomal RNA seems to be the most basic of our nucleic acids. Okay, so we've talked about the different types of RNA. Now let's go back and look at amino acids again. Let's review that. And I promise that I'm not just trying to throw words out here for no reason. I want to review what all these things are because then we're going to talk about how these players interact. So just hang with me for a little bit. Okay, so amino acids. If you'll remember in chapter 3, we learned that there are 20 different types of amino acids. And those are just the building blocks or monomers that we put together to make proteins. And so one of our examples here is glutamine, for instance. That is one of the 20 amino acids. And remember, when we link those amino acids together, that is the primary structure of a protein. And so it's going to be important, the chemical nature of these various amino acids, because how they interact is then going to determine the secondary and quaternary and tertiary structures of these proteins. So right now, I just want you to remember that there are 20 different types of amino acids. They can be used in all kinds of different orders to make an array of various proteins. So this is an overview of how we make proteins within the cell. And there are 2 different parts to making proteins. One is transcription and one is translation. So we begin with our DNA, which we know codes for all of our proteins in our cell. And what transcription is basically unwinding that DNA. And we have our template strand and our complementary strand. And remember, this is all taking place in the nucleus. And what happens is we then create a strand of messenger RNA that is complementary to our DNA template. So we have our C that lines up with our G, our adenine that lines up with our T, and remember, we don't have T's, so we have a uracil that lines up with our adenine, again our guanine with our cytosine, our cytosine with our guanine, and again with our guanine. And here we have 3 uracils that complement the adenine, because we are making a complementary strand that temporarily binds with the DNA template. And remember, those are hydrogen bonds, easy to break. Okay. Each of 3 nucleic acids make up what we call a codon. And that codon, CAU, GCC, UUU, each one is specific to an amino acid. Now, having said that, there are multiple codons that code for the same protein. So there is redundancy within our system so that if we have any damage, we can still produce the proteins that we need to. So we've done transcription. Transcription is taking DNA and creating a messenger RNA, and all that happens in the nucleus. Then the messenger RNA is sent to the ribosomes. And this next portion is what we call translation. So it's basically saying, okay, here's our code, we're going to decode it and make a protein from it. So like I said, each one of these codons then codes for a particular amino acid, and they're made in order along the mRNA, and they code for a particular amino acid. So CAU, for instance, codes for histidine. This is one of our amino acids. And so we denote amino acids usually by a 3-letter grouping. So HIS is for histidine, ALA is alanine, PHE is phenylalanine. Okay, so this again is translation, and it takes place in the ribosome. Okay, so here are the ones that we just, the amino acids that we just produced. Okay, so here is our phenylalanine, our alanine, and our histidine. Okay, so they have specific chemical structures, and that's going to become important. So this should be a little bit of a review. Here's our primary structure of our protein, and that's what we have just produced. Each of these little beads represents one amino acid. So we can pretend in here that we have our histidine, alanine, and phenylalanine, and so that's our primary structure. Now, based on those chemical formulas of those amino acids, then we get our secondary structure. And remember, that can be an alpha helix or it can be a beta pleated sheet. From there, then we go into our tertiary structure, and that has to do with all kinds of different chemical groups within our amino acids and how they interact with each other. That can then have our tertiary structure, and then multiple ones joined together for a quaternary structure, if that's going to be the destiny of that particular protein. So when we look at the codons always have specific amino acids, like I said. And there are certain codons that are start codons and stop codons. A start codon means, okay, you need to start making the protein here. This is where the code is written for the protein that we're looking for. And this table right here gives you a whole bunch of codons and what they code for. No, you don't have to memorize all of those, but I do want you to know the start codons and stop codons in particular. So if we look on this chart and we are looking for start codons, we find that there is only one. So that is actually, let me see if I can get my pen here. It's right here. AUG. And that's the mRNA. So our nucleic acid, A, U, and G, adenine, uracil, and guanine. That is always the start codon, and there's only one of them. And that start codon always codes for the amino acid methionine. So every protein starts with a methionine amino acid. And then there can be a series of codons, depending on how long the protein is going to be, how many amino acids are in it. And then there's always a stop codon. and it can be one of any of these three, UAA, UAG, or UGA. Those are the stop codons. That means it tells the enzymes that are involved in producing these amino acids to stop reading right there. And a lot of times they knock it off. They knock that particular protein off. But those are our stop codons, UAA, UGA, and UAG. Okay, now you have a lot of the terminology. So let's go back to, we said gene expression has two stages, transcription and translation. Transcription is making messenger RNA from the DNA, and translation is making proteins from that mRNA. Okay, so here is just another overview, a little more detailed. We have our double helix. It gets unwound, and we have the template DNA right here with its bases sticking out. And then this is transcription in the nucleus. We then have complementary mRNA. And note that each of the three pairs is a codon. And each codon, each three base pairs, then is translated at the ribosome into a particular amino acid. In this case, we have arginine, tyrosine, and tryptophan. And that, when they're bound together, makes a polypeptide, or a protein. Okay, so gene expression. Making a protein from a gene, we said, has two parts, transcription and translation. The transcription part is taking the DNA gene and making messenger RNA. So let's take a closer look at that. That right there is going to require an enzyme called RNA polymerase. Now that might sound familiar because when we were replicating DNA, we used a protein called DNA polymerase. So similar functions. The RNA polymerase is going to untwist and unwind the DNA. It's going to recruit the free nucleotides to the DNA and basically make an mRNA strand based on the DNA template. Okay, so let's take a closer look at the DNA gene itself. Okay, when we unwind our DNA, and this is our lovely DNA here in white. I know it doesn't look a thing like DNA, but use your imagination a little bit. We have, first of all, what's called a promoter sequence. The promoter has a sequence of nucleic acids that's found right before the part of the gene that needs to be transcribed. And so the promoter is the area that tells the RNA polymerase where to start. And that's actually really important because if you think about how our genes are coded, it's a sequence of 3 nucleic acids at a time. Now, if our RNA promoter is off and it starts on, say, the second nucleic acid, well, that can mess up the entire series. So this promoter portion is actually really important. So the promoter is where the RNA polymerase begins. Then there's the transcribed region, and that's the DNA sequence that the RNA polymerase reads and uses to make the mRNA strand. And then there is what we call a terminator sequence, and that is exactly what it sounds like. It's a DNA sequence that comes after the transcribed region, and that tells the RNA polymerase to stop reading the genes right there. So that's sort of an overview of what our DNA looks like for the part that we are going to transcribe into a protein. So here is our overview again. We have our DNA, this lovely stringy thing that for some reason in this image looks like an earthworm. I don't know why, but okay, there's our DNA sequence, and we have our promoter right here that tells the RNA polymerase that it can start transcribing. We have our transcription region and our terminator, and that's transcribed into a messenger RNA strand. Now this image also mentions what we call a regulatory sequence. And basically that is just a place that regulatory proteins bind, and the role of those regulatory proteins is to basically determine how fast this region is transcribed. And it doesn't always have to be right after the promoter. It can be throughout the transcription region. But if you remember the importance of homeostasis within a cell, these regulatory proteins play a role in that, because if you need, for instance, to produce insulin and you need it at a high rate, those regulatory proteins can come in and say, okay, let's make this faster. We got to get more of these produced. Consequently, if we have plenty of insulin, oftentimes the insulin will bind a regulatory protein that together binds the regulatory sequence and says, okay, stop making this. We don't need so much anymore. Okay? So that's what that regulatory sequence is for. So here's our picture of our RNA polymerase. That's that big enzyme that's going to transcribe the DNA to messenger RNA. And so in this particular image, our blue DNA is opened up, and we have the template strand on the bottom here. And in this case, the complementary strand really doesn't do anything. We don't need that right now. It's just sitting there as a placeholder. And so RNA polymerase then begins to build a messenger RNA by recruiting these RNA nucleotides, and we'll talk about a little bit more about that, to come in. And for each codon, so we're putting in complementary messenger RNA. And so remember, each of these three is going to be a codon that's then going to recruit an amino acid. But right now, the RNA polymerase is making new messenger RNA, and it does have a direction of transcription. So here we see that RNA is built in the 5' to 3' direction, just like when we did DNA replication. Okay, so that's always going to be the case, but for this time, remember, we're using only the complementary strand. So if we aren't using that other strand, then we don't need to worry about things like Okazaki fragments like we did with DNA replication, because we're only using one strand of the DNA, and we can make it in make our messenger RNA in a 5' to 3' direction, nice and smooth. Okay, so big picture, when we are creating proteins, remember we said we have transcription process and translation. Okay, now we are going under transcription, and there are 3 steps to the transcription part. And this is pretty easy. It's pretty intuitive. We have initiation, elongation, and termination. And so what that means is the initiation step of transcription is when the RNA polymerase finds the promoter and begins transcription. The elongation step to transcription is basically when the mRNA is being made. Okay, so it's being transcribed. And then the third is the termination. The RNA polymerase reads the terminator sequence and falls off the DNA template. Okay, so that's the 3 parts to basically making messenger RNA, which is what transcription is. So just to show you how important that one enzyme is, we have poisonous mushrooms called the death cap mushrooms. And so this is why it's important when you're going through the forest. You know, some people go, oh, we can eat mushrooms, and ha ha ha, we have hallucinogens, and okay, that's lovely, but what about you knowing your mushrooms? Because death cap mushrooms inhibit the activity of RNA polymerase within liver cells. And they do that by binding the RNA polymerase in a way that means that the RNA polymerase cannot bind the DNA. So if we're not binding DNA, we don't have transcription occurring, and therefore we are not making the appropriate proteins within the liver cell. And so people who eat the death cap mushrooms tend to die within 10 days because this one protein is inhibited. So that should tell you the importance of RNA polymerase. Okay, so what is the end result of transcription? The end result of transcription is an mRNA that's like a rough draft. It's called the primary transcript. And it goes through processing, RNA processing, to fix that rough draft. In other words, it's going to find out if there are any mistakes and fix those. And that only happens in eukaryotes. And remember, we had this very similar thing with our DNA polymerase, and that processing of DNA only happens in eukaryotes. So aren't we glad that we are eukaryotes? Okay, so there are 3 steps that go into this RNA processing. So the very first thing that's going to happen to the messenger RNA is an addition of what we call a 5' cap. And so that is an addition of a guanine triphosphate molecule added to the 5' end. So what does that do? Okay, that cap allows the ribosome to recognize the messenger RNA. In other words, the ribosome that's going to produce the proteins has to be able to recognize our messenger RNA that comes out of the nucleus. So that 5' cap is what allows the ribosome to recognize it. The second thing that mRNA processing involves is the addition of what we call a poly-A tail. And so that is put onto the 3' end of our messenger RNA, and it's anywhere between 150 to 200 additional adenines. Why would we do that, right? Well, the function is that it helps the messenger RNA get out of the nucleus and keeps it from being degraded. Okay, so it's sort of like a signal part of the messenger RNA that takes it out of the nucleus and protects it from being degraded. And then there's the third part of RNA splicing, and this I find completely fascinating. So when we look at the messenger RNA, it has what we call exons and introns, different portions throughout the messenger RNA, and you can see those pretty well in our diagram over here. So we have exons and introns, and the exons are the parts of the messenger RNA that we keep. Those are the ones that are what we call expressed or translated into amino acids. And then the introns are the parts that are going to be removed. It's almost like they're fillers within the messenger RNA, and those are excised out. We take those out of the messenger RNA. Now, just something fun to think about is, why do we have introns at all? I think it's a great question. I don't know the answer. I don't think anybody knows the answer. Introns have always been thought of as portions of the messenger RNA that are just junk and thrown away. That's lovely to think of, but the highest organisms all have introns, and those lower organisms, the way we think of it, lower organisms, like our bacteria, don't have introns. So I feel like they must serve some purpose that we just don't know about yet, which is always the exciting part of biology, is that there's always something new to learn, because even at the cellular level, there's so much we don't know yet, which keeps it exciting. Okay, so we are going to take away those introns and remove them. What removes those introns? Lovely name. I always love how these enzymes are named. They're called the spliceosomes, and it's actually more than one protein put into a complex of proteins that recognize those intron sequences and cut them out, and then they splice together the exons. So it has to be a multi-protein complex, right? Because that's a lot of functions. So we call that the spliceosome complex. Okay, so here we go again with our worm-looking DNA. We have our promoter right here. We know that's where our RNA polymerase starts. And then we have our transcribed region, which is full of exons and introns, and then we have our terminator region. And we know somewhere in here we also have our regulatory sequence that our regulatory proteins bind as well. So in transcription then, remember this happens within the nucleus, we create a messenger RNA or a rough draft of the messenger RNA in the 5' to 3' direction, and it still has the introns in it. And then we go through the processing. Step 1, we cap the protein. Step 2, our spliceozymes come in. And step 3, we put a poly-A tail on it. So that at the end, we have a 5' to 3' messenger RNA that has a 5' cap, a 3' tail, and only exons within the messenger RNA. Okay, so here's our summary again of our overview on making proteins. We've just gone through the transcription part that all takes place in the nucleus, and at the end of it we come out with messenger RNA that has had the introns removed, a 5' cap put on, a poly-A tail put on, in order for it to be able to go from the nucleus out to the cytoplasm. And interestingly enough, I didn't mention before, all of those RNA processing steps, they help to basically allow for the mRNA half-life to be much longer than those in prokaryotic cells. So it can last for several hours in the cytoplasm. Okay, but the next part that we're moving on to is translation. Okay, so translation happens in the ribosome. So we are taking mRNA codons and producing amino acids in order to line them up to make a protein. So the translation portion is when we take the messenger RNA and from it make a protein. So this is taking place in the cytoplasm with the ribosomes. So ribosomes are in our cytoplasm. And it involves our second type of RNA, which is tRNA, and which stands for transfer RNA. So far, our messenger RNA has basically looked like a straight line that was complementary to the DNA. But RNA doesn't always have to be in a straight line. It can be folded and have a U shape. And in fact, that is what tRNA looks like. So this is an example of tRNA. So even though it's called transfer RNA for the function that it provides, it's also helpful because it looks like a T in its structure. And so it has several different portions to it. So let's look at that real quickly. We have an amino acid at the end here, usually leucine, and through hydrogen bonding, it will complement with itself to do some folding like this, right here and right here, and then also down here. And then it has a portion called an anti-codon end. And that's something that should be somewhat self-explanatory. We've talked about codons in our mRNA that is a series of 3 nucleic acids, and so the anticodon is also a series of 3, but they are complementary to our codon. So each transfer RNA recognizes one mRNA codon, and it recognizes it by using the anticodon, which is that complementary sequence. So the tRNAs are out there in the cytoplasm. They recognize the mRNA with their anticodon, and they carry the amino acid indicated by that messenger RNA codon to that mRNA site. So when we look at the bases that we talked about, we have adenine, guanine, cytosine, and uracil, right, for all our RNAs. So every possible combination of 3 that we use those for gives us 64 different codons. And for those 64 different codons, there is one tRNA for every possible mRNA codon. So they're very specific. Okay, so if these tRNAs are in the cytoplasm and they have to bring an amino acid over to the messenger RNA, how do we know, or maybe I should not how do we know, what makes sure that the tRNA is attached to the correct amino acid? Well, that is the role of what we call amino acyl tRNA synthetase. I know that's a mouthful, but it's an enzyme that makes sure that the amino acid is correctly placed with the right tRNA. And that is a process that involves ATP, as you might expect. So it basically makes sure that we have excuse me, an amino acid coming in, and of course as an enzyme it has a particular binding site. ATP does as well, right? And then we have the tRNA that comes in and binds, and so then it attaches the amino acid to the tRNA and then it's released from the enzyme. And so that is how every amino acid is attached to the correct tRNA. And that enzyme is called amino acyl tRNA synthetase. Okay, so here's our overview again on making proteins. We've done transcription in the nucleus. We've come up with the mRNA that has been corrected and sent out into the cytoplasm, and then we are now looking at translation in the ribosomes to make proteins. Okay, so we said there's at least one tRNA for every amino acid. So you can picture that as our codon on our mRNA with our 3 nucleic acids, the anticodons on the tRNA, and then here we have the amino acid that's been attached. And so as we go down the mRNA, we go to each codon and have the tRNA attached. When the tRNA is full of its amino acid, we call that a charged tRNA. So when it brings in the amino acid, the charged tRNA binds the codon on the mRNA. So here we are just looking at it overall. Our transcription, we had our template strand of our DNA, and then our messenger RNA that was created in the 5' to 3' direction. And from that we have our anticodons that come in and bind to the codons in the messenger RNA, remembering that the start codon always is coded for methionine right here. Okay, so these are charged tRNAs that are binding to our codons on our messenger RNA. So this is what it looks like because this is happening on our ribosome. So our ribosome, we have the mRNA right here in the small subunit, and then our transfer RNA comes in on the large subunit, and our peptide chain is created. Now, let's take a look at our ribosomes, and because it has different sites that I want you to know about. Okay, so like we said, the ribosome comes in a large subunit and a small subunit. So let's start with our A site. Our A site on the large subunit is where the tRNA first arrives. So it first comes in, so you can use arrives for the A site. The P site is where the tRNA is holding the polypeptide or the amino acid chain. And then the E is the exit spot for the tRNA. Okay, and this, of course, down here is where our messenger RNA is going to be. Hopefully that makes sense to you. So there are 3 steps of translation, just like there were 3 steps of transcription, and amazingly they are the exact same. So initiation, elongation, and termination. So in initiation, that's when the first tRNA carrying the methionine is called, and that's called the initiator tRNA. Because remember, that's where the start codon is. So here's our, let me get my pen. Our start codon, AUG, which codes for methionine, so that begins our translation. So it's initiated right there. Our second step is elongation, and just like you would expect, we have multiple tRNAs coming in, and we can have 2 tRNAs at one time on our ribosome, so they can both be acting at the same time. And so our amino acids come into the A site, and then they're going to, of course, leave the exit site. In the meantime, we have our amino acid chain beginning to grow, and that's our P site, remember, for polypeptide. Our step 3, of course, is going to be our termination. So a protein comes in to make sure that it's terminated, and that's called a release factor. And that release factor recognizes one of the 3 stop codons, and it stops that translation. So when it does that, then we have a release of our polypeptide, which is, remember, our primary sequence of our protein. Okay, so let's say that this is the polypeptide that we are creating. So if we take a look at this, we've started with our start codon, of course, which is going to put in our methionine amino acid. And then we have all these other amino acids that are then ligated together to form our polypeptide. And oftentimes one or a few of these amino acids are used as a signal to send our polypeptide from the ribosome on to the next area where it's going to get processed. So this is going to look familiar based on what we've talked about before with the different organelles and what they do. So here is our rough endoplasmic reticulum. This is the primary place for protein synthesis. It can also happen in free ribosomes within the cytoplasm. But there are so many ribosomes on the rough ER, that's where most of our protein synthesis occurs. When it is completed, oftentimes we have transport vesicles that then take those proteins to the Golgi apparatus. And remember, one of the functions of the Golgi apparatus is to modify proteins from the endoplasmic reticulum. So that's where we start all the folding processes, and all those additional enzymes that make sure that the proteins get folded properly are in the Golgi apparatus. Once they're all folded properly, if the protein is going to be used outside of the cell and in a different location, then it is then packaged in one of the secretory vesicles, and then through excytosis it is sent outside of the cell. And that is the destination of our protein that started with our DNA and transcription through translation, modification in the Golgi apparatus, and sent out. So here is the summary of gene expressions in eukaryotes. So we start with our DNA in the nucleus, and that serves as a template for our messenger RNA. So the messenger RNA is created, and then before leaving the nucleus, remember it goes through those 3 steps of being processed, then it leaves out the nuclear pore into the cytoplasm, and either in free ribosomes within the cytoplasm or ribosomes on the rough endoplasmic reticulum is where translation occurs. And so that means that the tRNAs with their anticodons are brought in to match the codons of the messenger RNA, and when they are charged tRNAs, they bring with them their amino acids that can then build the polypeptide chain. And so polypeptides are synthesized, and then they are sent on to the Golgi apparatus, where they are modified, folded correctly, and then they can either be used within the cell or sent out through exocytosis. So the major differences between prokaryotic versus eukaryotic gene expression is that in prokaryotic cells, we have transcription of the DNA straight into RNA. There's no nucleus, so we don't have to worry about going through a nuclear pore or anything like that. the mRNA is made, there's no pre-mRNA, it's just straight mRNA because there's no modification to it. So once the mRNA is made, it's directly translated into a polypeptide by a ribosome. Our eukaryotic cell, however, transcription is still taking place, but it's taking place within a nucleus. We make basically our rough mRNA or pre-mRNA, which is then processed into mRNA, and then it is released through our nuclear pore into the cytosol where it's translated at the ribosomes. So when we look at it in comparison, gene expression in prokaryotes happens entirely in one place, whereas in eukaryotes, the transcription happens in the nucleus with translation happening in the cytoplasm. In the prokaryotes, the RNA is not processed, whereas it is in our eukaryotes. And in prokaryotes, transcription and translation occur at the same time. I didn't mention that before, sorry. So because it's all in one location, it all happens on what we call polyribosomes. So transcription and translation happening at the same time. In eukaryotes, the transcription and translation are separate events in separate locations. So here is an image of that polyribosome that we were talking about. So in this image, we have our DNA right here. So you can actually see it in the micrograph right here. There's the RNA polymerase. And then as the mRNA is being produced, the ribosomes then attach to the mRNA, forming this polyribosome unit, and then proteins are produced right there, all happening in one place at the same time. Okay, so let's talk about if we have problems within our DNA that aren't corrected. So the top left here represents what we would expect with our DNA. The complement is correct. So we talked about before that we could either have deletions, additions, or base substitutions that could be problems. So how does this play out through gene expression? So each of those is called a point mutation. That means that there's one change in a nucleotide sequence. And that seems like such a small thing, but it actually turns out to have pretty large consequences, which that in itself ought to blow your mind for how many times we have gene expression and that it's done correctly. That by itself should set you in awe. Okay, so deletions, additions, and base substitutions are all examples of what we call point mutations, one change in a nucleotide sequence. So in the next slides, I'm going to show you what can happen with a point mutation. And specifically, we're going to look at the disease called sickle cell anemia. Okay, so our normal hemoglobin, so that hemoglobin is that protein in the center of your red blood cell that allows you to carry oxygen to all parts of your body. So as we go along in our DNA, and we have CTT as one of our codons, and then here are our anticodons, GAA, which codes for glutamine. What happens in our point mutation, let's say that this one here in the middle gets thrown off, and instead of CTT, we now have CAT. Well, that changes the anticodon then to GUA. That GUA codes for a totally different amino acid, valine. And the result is a sickle-shaped red blood cell because we have changed the hemoglobin protein. So what we call normal DNA, the other term for it is what is called wild type. We have wild type versus mutant hemoglobin. So here we go with our wild type DNA at the top, with the mRNA being processed just the way it should be. And then here is our mutant hemoglobin with our substitution right here of A, and that provides a valine as opposed to a glutamine. And this is actually a micrograph of what your red blood cell looks like. It's not nearly as functional. So the interesting thing is that if we have a base substitution at the DNA level, at the protein level that can result in 3 different types of mutation. So when there's no mutation, for instance, again we have our DNA TTC, mRNA level complementary would be AAG, and that results in a lysine amino acid. So with these, if we have a point mutation on the end and we substitute the cytosine for thymine, our mRNA level is going to be an adenine instead of a guanine. So interestingly enough, AAG and AAA both code for lysine. So this is what we call a silent mutation. So you wouldn't notice a difference on that one because it still codes for the same amino acid. Now, if we had a point mutation at the beginning of this codon and we replaced the thymine with the adenine, our mRNA would be a uracil at that point then, and the codon would read UAG. And that would mean a stop sequence. So our protein would stop being made at that point, in which case we really just have a part of a protein and not a full protein. So that's what we call a nonsense point mutation. A missense then is this next point mutation, and let's say we substitute our thymine for a cytosine. That would result in a guanine in that point for the messenger RNA, and AAG codes for arginine. Well, that will mean the wrong amino acid. Additionally, we could have replaced it with a guanine. The mRNA level would have been a cytosine, and then we're coding for yet another amino acid. So that's the wrong amino acid in that particular sequence, which may at the primary structure not quite look bad, but by the time you start getting into secondary structure and tertiary structure, all of these chemical bonds make a difference in how the protein is folded, and so we're going to have the wrong protein. So the way we can look at this is with the DNA substitution, we have a silent mutation where the nucleotide change doesn't affect what amino acid is coded for, a nonsense mutation where the nucleotide change codes for a stop codon and chops the amino acid sequence, or we can have a missense mutation where the nucleotide does code for the wrong amino acid, completely messing up the protein. So hopefully that explains then how with sickle cell, when we have a point mutation, it codes for the wrong amino acid, and therefore the hemoglobin protein does not fold correctly. So this is a missense mutation. A fourth type of mutation is at the DNA level is when you have either an addition or a deletion. And at the protein level, this can result in what we call a frame shift mutation. So if our wild type or normal sequence of DNA looks like the frame on the top, and it gets read, if you think about this as a codon, each frame being a codon, and it would be read just like that, then if we add or delete a particular nucleotide, then that throws off the codon. So now we would be reading across like this, right? And so that is called a frame shift mutation. So this is what it looks like if we're looking at our DNA. Our normal or wild type DNA. Here we have our codon and our anticodon. So for here, we are making a protein that has lutein, alanine, cysteine. But when we have our frame shift, we've added a guanine in here. And so whereas we're normal here until we get to this addition, now it shifts and we have arginine, methionine, and that's not what this was supposed to be coded for. So that's called a frame shift mutation. So it's an addition or a deletion of a nucleotide that changes the order of 3's. Okay, so that is your transcription and translation for gene expression, and then how that can go awry. Okay, so that is it for our gene expression. Make sure you review that and you know again all the players and what can go wrong.